KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.com.
Students, now in this video we are discussing continuation information of inducible Operon concept. So under inducible Operon concept, so up to regulator gene we have discussed now. So inducer gene information I didn't give you yesterday. So by covering that particular information, so here on NEET examination point of view, somewhat important concept I will explain in this video. Okay, sorry, right. Now let us see inducible Operon concept. So under last and final side heading under this concept, that is inducer. So by seeing the inducer, so you may understand, okay, we are going to discuss regarding lactose. So why? Because the lactose only acting as inducer, so thereby, so inducer is nothing but lactose. Now only don't consider inducer as a lactose, just generally you consider inducer. Now once let us see, inducer is nothing but it is a chemical substrate. Inducer is a chemical substrate or hormone or some other metabolite. So whatever it is we can consider. So inducer, that we can take any kind of or chemical substrate or hormone or some other one particular kind of metabolite we can consider. So which after coming contact with the repressor forms an inducer repressor complex. So means here when this particular inducer will come into the contact with one repressor, automatically which one formation takes place? Inducer repressor complex formation takes place. Means once you see there in presence of lactose, we draw no lactose is there, that particular lactose will go and binds to the repressor protein. When this binds to the repressor protein, the structural and the functional activities of repressor protein will be changed. So thereby repressor protein cannot be binds to the operator. Likewise we learn no, so adida. So means here when this particular inducer will go and get contact with the repressor, so thereby which one formation takes place? An inducer repressor complex formation takes place. Now this complex cannot bind with the operator gene. So correct only no. So when this entire complex repressor gene with the inducer, when these two combinedly forms a complex, that particular complex will not or cannot bind to the operator gene. So when this entire complex will not be connected or will not be attached to the operator, no, so definitely this particular operator gene will be switched on. Switched on means that at the time that will be activated. Okay, means when this repressor protein cannot bind to the operator, so that time only we will observe the activation or the switch on mechanism of operator gene. Why? Because at the time we see no, when operator gene location is free, at the time no, the pathway of RNA polymerase is free. So likewise, so thereby what is the enzyme or that is maybe RNA polymerase enzyme present at the promoter region starts to move towards the structural genes we see no. So means what is the point here we have to understand no, when repressor protein, listen carefully, when repressor protein cannot bind to the operator gene, no, then only operator gene will be switched on. Correct only no? Right. Next one, the free operator gene Once you see the free operator gene allows structural gene. One more time, the free operator gene allows the structural genes to transcribe mRNA to synthesize the enzymes. So means here when that particular operator gene will be switched on. So means switched on means when that particular operator gene must be free, isn't it? So when free means that one when it is not binded by the repressor protein, isn't it? So thereby when free operator gene allows the structural genes to transcribe the mRNA to synthesizes the enzymes. So means here structural genes definitely transcribe. Structural genes definitely transcribed into the mRNA when only operator gene will be switched on or operator gene is free. Point puriyadana. So now here what is the point we are discussing? Definitely operator only, definitely entire work keeps on its shoulders. Means here what is the point? Definitely here the operator should not be binded by the any kind of repressor protein, then only that particular operator gene will be activated. So thereby means here operator location must be free, then only it can work effectively. Point puriyadana. So next one, now you concentrate. Now inducer, next parapathama. In this particular thing, now we are considering inducer as lactose in case of E. coli. So one more time you see, inducer for lack of operon of E. coli is lactose. Means here what is the point now we are taking in E. coli point of view or in prokaryotes point of view, what are the main important points we are discussing here we didn't discuss, means we didn't give name as inducer is nothing but lactose kandipa ade edutunga, other than that nothing, no need to take or no need to take anything, likewise we didn't discuss. So overall means that particular inducer may be chemical substrate, maybe a hormone, maybe or some other kind of metabolite we took, but for E. coli point of view, so Escherichia coli point of view, so inducer what we are taking for lac operon concept point of view, we are taking inducer as lactose. Lactose we are taking as inducer. Okay, so thereby here in lac operon concept, lactose we are considering as inducer. Okay, now here what is the point? In fact, allolactose an isomer of lactose. So this one we know very well. So here conversion of lactose to allolactose, so that is under the control of beta galactosidase we see now. So two functions we see now. What are those? First one, beta galactosidase that converts the lactose into glucose and galactose. That is first function. Next work of beta galactosidase is that converts the lactose into allolactose. Okay, so adi pakkathali podunga. Ipo what is the point? In concern of lac operon of E. coli, inducer is nothing but lactose. Now let us move forward. So when the sugar lactose is added to the culture medium of E. coli, a few molecules of lactose gets into the bacterial cells by the action of enzyme nothing but permease. My point I think so we understood. Means here we are keeping or we are growing bacteria or E. coli bacteria in one culture medium. That particular culture medium has or having which one? When the particular culture medium having of lactose, so some amount of lactose will be entered into the bacterial cells because of the activity of permease. My point purida means so definitely what is the culture medium consist of lactose in that particular place. So suppose if you keep E. coli, so with the help of permease enzyme, these particular lactose molecules translocated to the bacterial cells. So thereby here, so simply one more time let me give you. permease enzyme will be there in the bacteria. So because of the activity of a permease enzyme, so what are the lactose molecules are present outside of the bacterial cells now get converted or get translocated into the bacterial cells. Means here the transport of lactose molecules, everything is under the control of permease. So now here one important note we have to discuss. அது என்னன்னா இப்போ பாருங்க இங்கே a small amount of this enzyme is present in the cell even when the operon is not working. Point புரியுதமா? So here definitely a small amount or the small quantities of permease enzyme is present in the bacterial cell even though that particular entire operon is not working condition. At least a little quantities of enzymes definitely will be there even though that particular operon is under not working process or not progressing process. Okay, so now here these a few, once you see these a few lactose molecules are then converted into an active form. You see there now what are the lactose molecules? What are the lactose molecules are entered into the bacterial cell and these lactose molecules are then converted into an active form. So now when these lactose molecules are get converted into the active form which acts as an inducer. So definitely what are the lactose molecules are entered into the bacterial cell, these particular lactose molecules are converted into active form, so thereby and eventually acts as inducer. And when this particular inducer's formation takes place with the activity of lactose molecules, so definitely binds to the repressor protein. So now here what is the thing? Definitely this particular inducer means lactose will go and binds to the repressor protein. So when this particular lactose will go and switch or binds to the repressor protein, no, so simultaneously what is happening? The activation or till now the repressor protein is activated, but when the inducer will go and binds to the repressor protein, automatically the activated repressor protein will be inactivated. So thereby it cannot be binded or it cannot binds to the operator region. So thereby that is only, let us see now the inducer repressor complex. Once you see inducer repressor complex means ange yaar irukku, one is inducer is there and one more is repressor protein is there combinedly. So that particular complex we are calling as inducer repressor complex fails to join with the operator. So means when this particular inducer repressor complex is there, in that particular case no, that entire complex will not be connected or will not be joined with the operator. So thereby operator region will be turned on. My dear students, one point nalla save panikonga, only when the repressor protein will go and switched or when will go and binds to the operator, no, then only operator mechanism will be turned off or switched off. But here inducer and the repressor, when these two are combined, no, at the time inactivated complex formation takes place. At the time no, inducer repressor complex will not go and will not binds to the operator. At the time definitely we will observe the switch on mechanism of operator gene. So thereby operator gene is turned on. So when operator gene is turned on, so then what is happening, sir? Means once you see the three genes are expressed as three enzymes to metabolize the lactose. So definitely here what are the main important points here we are discussing means, so in presence of or when the operator gene is active, so thereby the pathway of RNA polymerase is free. And here our knowledge point of view, here the RNA polymerase enzyme is present at the where promoter region. So when the pathway of RNA polymerase is free at operator region, definitely RNA polymerase enzyme come across with the operon and finally reaches to the structural genes and definitely involves in the transcription and translation processes and definitely involves in the synthesis of Three different types of enzymes, nothing but beta galactosidase, permease and transacetylase, which are produced from the ZYA genes respectively. So here what are the genes are produced? Emma, what are the genes are produced from the ZYA genes, or sorry, what are the enzymes are produced from the three different types of structural genes? No, definitely necessary, definitely necessary to metabolize the lactose. Means here in presence of these kind of enzymes only definitely we can take this lactose into the metabolic activities. Why? Because what is the lactose? Now it is entering into the bacterial cell. Now that must be converted into another forms. Why? Because suppose in the presence of beta galactosidase, this particular lactose must be converted into glucose and galactose. Emma. So now here one point you listen carefully. Ipo this up to here normal da. Next one this point pathukide. Allolactose is a real inducer of lac operon. Emma, one more time you see, allolactose is a real inducer of lac operon. Means here main important point, lactose is there, that particular lactose in the presence of beta galactosidase. Emma, lactose is there, that particular lactose in the presence of beta galactosidase enzyme that will be converted into allolactose. Now here we are discussing, no, here the lactose will go and binds to the repressor protein. So that particular repressor protein will not be binded to the operator we are discussing, no, adi pakkathali pudinga. Ipu perfect one and the real inducer of lac operon is allolactose. Allolactose is nothing but isomer of lactose only, no, avalada. Why? Because these two means lactose and allolactose both are isomers. Now here one important name I would like to give you. That name pathukuma gratuitous inducer. Gratuitous inducer. So underline pannala but no try to save pannikunga. Gratuitous inducer. So what is the meaning and what is the important concept to be discussed under this is some molecules resemble with the natural inducer but are not metabolized by the enzyme. Once you see gratuitous inducer means that is also one kind of inducer only. That is also like lactose madirda. But these molecules seems to be resemble similar to natural inducer means eppadi lactose or eppadi allolactose is there, no, even gratuitous inducer also being like that only but are not metabolized by the enzymes. Means here that particular inducer cannot be metabolized by the enzymes. Three different types of structural genes will be released, no. So all these structural genes cannot be metabolized metabolize this gratuitous inducer. But gratuitous inducer seems to be like a normal inducer only, but what are the enzymes are there or what are the enzymes are produced from the structural genes? These structural genes cannot do anything to that particular gratuitous inducer. Means here this particular gratuitous inducer cannot be involved in metabolic activities. Okay, for example, what is the enzyme? No, isopropyl thiogalactosidase. Or once you see isopropyl thiogalactosidase or galactoside. Okay, isopropyl thiogalactoside. So this one simply we will be considering as IPTG. Once you say that particular enzyme name or that particular metabolite name or the molecule name, that is isopropyl thiogalactoside. So simply IPTG. So this particular IPTG so resembles lactose and thus has a property of induction. It seems to be like a lactose and definitely it has a property of inducer. And the phenomenon is nothing but induction. So such inducer which induces the enzyme synthesis but without getting metabolized. So means here, what is the enzyme or what is the inducer? Now we are observing now here, this particular inducer induces the enzyme synthesis. Means here there are 3 different types of structural genes. Definitely formation takes place, but this particular inducer getting or without getting metabolized, means here this particular inducer will not be involved in metabolic activities. So such particular inducer we will be considering as gratuitous inducer, and that particular inducer best example of the molecule name is IPTG. IPTG is nothing but idida. Okay, isopropyl galactoside. Now here இப்போ diagrams now new important concept this one not given even in our NEET examination also, NEET examination material, but this small concept I would like to explain. Okay, so now till now what is the information you learn no அது எல்லாமே பக்கத்துல போடுங்க, இது ஃப்ரெஷ்ஷா லேர்ன் பண்ணுங்க. Okay, now that particular concept is catabolite activator protein. One more time you see, catabolite activator protein, simply CAP. Emma, one more time, once you say along with me, catabolite activator protein. Emma, ipo one important concept adu enna, idu parunga, idu enadhu petri dish. In our biology lab once we see no petri dish, adhe round shape irukku. So once we gone to our laboratory and we see no petri dishes. So adida, so petri dish edthi hange material. Now here what are the sugar sources are there no? Once you see here this particular diagram, G is there no, G appadana glucose, L is there no, adi lactose. Okay, ipo orange color dots are there no, adi yarana E. coli. E. coli bacteria. Now we are growing E. coli bacteria in our laboratory conditions. So for example, now your teacher added which one no, glucose and lactose. Okay, ipo you have to get one doubt, sir, ipo sugar sources rendu irukudana, ipo glucose is there and lactose is there, appadana out of these two which one will consume first, sir, or which one will absorbed by the E. coli first, sir? Means amma point you understood era, means in comparison of these two sugars, one is glucose and lactose, no, glucose only first consumed by the E. coli. Emma, so nalla save pannikonga. So first of all, when there are 2 or more different types of sugar components are there in the medium, E. coli will absorb, or Escherichia coli prokaryote will absorb only glucose first. Means, sir, up to what time, sir, it will absorb the glucose? Means, so when this entire glucose molecules, when it is going to be completed, until then glucose is absorbed by the E. coli. Once the glucose levels are depleted, amma listen carefully, right? When the glucose levels are depleted, then only E. coli absorbs the lactose. Amma point purida, ipo two types of sugar components are there. Ipo E. coli, suppose if you are keeping into that one particular medium, that medium consists of two types of sugar compounds, so for example glucose and lactose. In this particular case, no, E. coli absorbs first only glucose. Means unless completion of glucose, lactose will not be consumed or received by the E. coli. First of all, glucose levels must be depleted, must be completed, then only E. coli absorbs the lactose. Emma, ipo save panninga, ipo koncham deep studies, our knowledge point of view. So if you see E. coli gene, you know, polycystonic gene or the E. coli Operon concept, genes are like this. PIPOZY we studied. Emma, idu ellarkume theriyum. But ipo ange koncham ulle, if you see deep studies at promoter region, amma see right here, in promoter region, cap binding region is there. Emma, in promoter region, which one is there? Cap binding region or cap binding site, whatever it is, idi cap site simply. Emma, idi cap site where it is there? Exactly at promoter region. Nalla save pannikonga. Okay, and our knowledge point of view, even RNA polymerase enzyme also is present, or RNA polymerase enzyme also will come and sit at the promoter region only, no? That particular point also save பண்ணிக்கோங்க. இப்போ let us enter into the actual concept. நல்ல important, listen carefully. Now, in absence of glucose, in absence of glucose or in presence of lactose, or simply, so we kept a side heading like this. What is it? Absence of glucose and presence of lactose. இப்போ பார்த்துக்கோங்க, absence of glucose we wrote, no, absence minus we kept, and lactose plus we kept, no. இப்போ பாருங்க, இப்போ explanation சின்ன பாயிண்ட் தான், பட் நல்லா சேவ் பண்ணிக்கோங்க. இப்போ in absence of glucose, what is happening, no? இப்போ in absence of glucose, listen carefully, the levels of CAMP will be more. That's why arrow mark we kept up, towards up. Okay, now you see there, so CAMP is nothing but cyclic AMP. In absence of glucose, the content or the percentage of the cyclic AMP levels will be more. Now the cyclic AMP levels, now we took as a chin dot. Once you see in brackets we kept one dot, no. Now in absence of glucose, what is happening, no? This cyclic AMP bacteria ulle poi, now it is there within the bacteria. Now this particular CAMP covered by the cap is nothing but, once you go up and here you see cap is nothing but catabolite activator protein. So when this particular catabolite activator protein covers the or binded to the CAMP, one complex formation takes place. Complex consists of catabolite activator protein and one more is cyclic monophosphate. These two will go and binds at the cap binding region or cap binding site. So this entire complex will go and binds to the cap region. So here we took, no, cap region or cap site. So when this entire complex will go and binds at the cap region, sir, what is the use, sir, of this one? Means at the time RNA polymerase will be activated more and more. So munadi, so we didn't see this one. So what we have seen, when repressor protein will not be binded at the operator gene, no, RNA polymerase enzyme will go and transcribe the structural genes we see. But if put deep studies point of view, what is the point we are discussing? In absence of glucose, CAMP levels, means cyclic AMP levels or more. Now these cyclic AMP levels in the bacteria binded by the or joined together by the CAMP. CAMP is nothing but catabolite activator protein. Now this one will go and binds at the promoter region. Why? Because promoter region consists of cap binding site, isn't it? So in this particular region, what is happening? No, because of this complex at the cap region, this entire complex will go and binds at the operator region. No, why? Because at operator region we are finding the cap binding site, isn't it? So because of this complex at the promoter region, RNA polymerase enzyme is activated more and more. Why? Because even RNA polymerase enzyme also is present at the promoter region only, no. So because of this entire complex, the activity of the RNA polymerase is activated. So thereby later all these genes or these three structural genes will be transcribed and translated simultaneously, and three different types of enzymes formation takes place at the time. Once the enzymes formation takes place at the time, so this particular E. coli will be activated to take the lactose. Point puridara. Why? Because here lactose also is there, no. So at the time when this entire complex, when these three structural genes transcribed and translated simultaneously, so this particular E. coli get ready, so to take the or to receive the lactose in that particular medium or present in the medium. Next concept you see, that side, in presence of lactose, sorry, once you see the side heading, in presence of glucose and the presence of lactose, inge rendume present. So in a first statement, what we have seen, glucose is absent, lactose is present, we see. But in a second statement, no, both are present. Glucose is there as well as lactose also is there. In this particular case, no, nalla save panikonga. Ipo glucose is present, we took no. Glucose levels also more. Once you see the pink color arrow mark turns towards up, no. So here the glucose levels also more. When glucose levels are more, so what is happening? The concentration or the percentage of cyclic EMP levels is less. Means here, did you understand one point? In first statement, when the glucose levels are less, so we have seen the percentage of cyclic EMP levels are more we seen. And when here second case point of view, when glucose levels are more, we are observing no, the percentage of CAMP levels are less. A point you understood no? So this is null save panikonga. So sometimes no, this particular point only they are asking as statements. Okay, now the glucose levels are more, so thereby cyclic AMP levels are less. Okay, now this cyclic AMP even though entered into the bacterial cell, here CAMP dot we took no, this CAMP and again, so here this entire complex, here entire complex of formed no, so this here in presence of glucose and presence of lactose, this entire complex will not be formed. So we drawn, but here what we have written, so this entire complex will not be formed. So entire complex when it will not be formed, no, it cannot go and it cannot bind at the promoter region. At promoter region we know very well, their cap binding region is there, no. When this particular cap binding region not consist of this entire complex, automatically the activation of RNA polymerase will be poor or is poor. Poor, what is that? So here RNA polymerase enzyme will not be activated more and more, like very slow dark. Okay, means here in absence of this entire complex at the cap binding region, at the time no, RNA polymerase activity will be very less. Sorry, RNA polymerase activity is less only, but functioning only no. Listen carefully, but functioning only no, at the time what is happening? So even though in less activation of RNA polymerase enzyme, so what is happening no? So koncha glucose molecules consumption process taking place. RNA polymerase is activated only no. So at the time no, when this particular for example, I will give you one example. Bike we are starting so now neutral bike start pani. If you leave means engine will be switched on mode only and we will not go anywhere. We cannot move forward, but entire petrol gradually will be depleted, isn't it? Likewise here what is happening? Even though RNA polymerase is less active, so here more glucose molecules are there now. Gradually glucose molecules levels are depleted. Okay, once the glucose molecules levels are depleted completely, at the time CAMP levels will be more. Are you getting? So at the time CAMP levels will be more. When CAMP levels are more, at the time this entire complex of formation takes place as left side explanation. So thereby this entire complex will go and binds at the cap region. Automatically RNA polymerase activation taking place more and more. Automatically this entire structural genes are transcribed and translated. Again, three different types of proteins and enzymes of formation takes place at the time. So this particular lactose consumed by the E. coli. So suppose no worry about this explanation. Suppose if you are unable to understand, so in presence of you I will explain this explanation by one more time. Okay, so thank you, be safe at home.